what happens. Oh, let me just make this clear. Sorry to go back. Codons from up here are AUG, CUA, and UGU. Okay. Now, there's another RNA that contains the anticodons. Okay? And I'm doing this one in green. It's transfer RNA. If you look in the notes, the transfer RNAs are larger than just 3 bases. So what we have here is something that looks kind of like a hairpin. I know that doesn't look really a whole lot like, let me get a little longer there. Looks more like a hairpin. Okay? And over here, in fact, I'd already said this is methionine. You don't have to remember that. You don't have to try to put all this together with the genetic code unless I have you do that in part 2 where you have a genetic code right in front of you. So, the base pairing rules still apply. And this is still RNA. So UAC is our anticodon. So our first anticodon is UAC, and that's because it base pairs with AUG. And that's how, this is so cool. This is how you get that particular amino acid in that place to make a protein that functions properly or that is different from another protein. This is so neat when you think about it. And oh, that genetic code I showed you, as far as we know, that thing's universal. That everything that is alive uses that genetic code. Now, if we have aliens among us, maybe theirs is different. But everything that we know of, every living organism uses that genetic code. All right, so now we have another one of these. What did I say it was? Was it leucine? I think it was. And it's the anticodon here is GAU. And it base pairs. So we've got this base pairing. Now this is a temporary base pairing. In fact, let's go back to that initiation, elongation, termination. I like to ask about the initiation of translation because there are 4 components that come together. The small ribosomal subunit, the large ribosomal subunit, the messenger RNA transcript, or our note card, and the first transfer RNA. If you think of these transfer RNAs as teeny tiny teaspoons, and instead of putting a whole cup of flour, you've got all these different types of flour, not different brands, different types, and you're putting them in 1 grain at a time. So this is the methionine type of flour. This is the leucine type of flour. And you're putting them in 1 at a time. And that's part of then after initiation where the messenger RNA, the first transfer RNA, and the 2 ribosomal subunits come together. That then what follows is elongation, the protein formation, basically putting this protein together. So, and I urge you to look at an animation where you see this actually moving, because that makes a little more sense, and I can't make it happen here. I can't make that magic of CG happen on a flat board. Okay, now our third one, ACA, and our amino acid here I think was cysteine. And what's happening is each time one of these, during elongation, one of these amino acids is being brought in, a bond forms between it and the previous amino acid, so that we end up with a protein that's got multiple amino acids. And finally, there's a stop codon. And when the stop codon is reached, that's termination. The two ribosomal subunits come apart, the messenger RNA leaves, and you have your brand new pre-protein. It's usually not ready. Okay? So that's translation. Couple of things. To add to this. First, this thing that happens that seems like a waste of energy in eukaryotes we have post-transcriptional processing. Somebody asked when it occurs. Post means after, and then you have transcriptional. So right after transcription, before translation, if this is the messenger RNA, and I'm going to do this with colors. So these are all RNA nucleotides along this, but it's pretty long. The green ones are introns. The blue sections are exons. And what happens is the introns are taken out. I know, doesn't make sense, right? You would think something called introns stayed in. Yeah, it's kind of like that oxidation reduction thing, right? But the introns come out, and we end up with our mature messenger RNA, and our pieces of introns are taken out, and our exon pieces are put together to make our mature messenger RNA from our pre-messenger RNA. All right? There is also post-translational processing that occurs after translation. But in that, and that would occur in prokaryotes as well, that's when the three-dimensional shape of the protein is made. So that long string of the amino acids, the primary structure of the protein, folds and makes a 3D structure. Okay? No, sorry, let me beep, beep, beep. It folds some and makes the beta pleats and alpha helices. But it doesn't completely form the 3D structure. So it'll have these little curly cues and these little pleated areas. And that's the secondary. This is in post-translational processing. Then all of that with hydrogen bonding, ionic bonding, whatever, forms the 3D structure of the protein. And finally, oh wait, not finally, we still have the cell cycle. Regulation. Okay, and I used to have people learn about the lactose operon that occurs in prokaryotes. But you know what? I don't want to try to explain that, especially in this online-only class. So I just want to say that regulation does occur because not all genes are being expressed or turned on at the same time. Or in multicellular organisms, not all genes are being made in each cell type. Let me explain that a little bit. The lactose operon, for example, in the prokaryotes. This is a way to save energy. So that in these lactose fermenting bacteria, lactose is a carbohydrate, when it is present and glucose is absent, those bacteria metabolize lactose. So, unless glucose is absent and lactose is present, that gene is not turned on. And that means that the messenger RNAs aren't being made and the proteins aren't being made. And hey, that saves a lot of energy for the cell because all of this takes ATP to happen. All right, so energy saving. And some of you wrote that down. Now I want you to think about the multicellular organisms. Think about the amazing organism that you are and how you have skin and how your skin is different than the muscle underneath it and how you have a brain with all these nerve cells and supporting cells of the nervous system and how those cells are different than your skin and your muscle cells. Okay, so think about that. Okay, and you even have different types of muscle and they're different from each other. How does this happen? This happens because different genes are turned on in different cells to make the cells what they are, to make the proteins that make the cells what they are. So if we made all of our proteins in all of our cells, we would be one big multicellular glob of the same type of cell. So we'd be like I forget his name, but he was on what's the sci-fi with, it was kind of a takeoff on Star Trek, what's the name of it? Orville. The Orville. So we'd be like that green guy that looks like snot. That's what we would be like, except we wouldn't have a mouth, okay, because that implies some differences. So yes, isn't this amazing that even though every cell that you have, whether it's a skin cell, a muscle cell, or a brain cell, has the very same recipes in it, inside the nucleus, that not all of those are being made so that those cells are different. This is amazing. It is. Oh, and again, there's some for saving energy as well, because there's no need to make a protein that's not being used. But a lot of it allows for, and let me put this in a short form, a diversity of cell types in multicellular organisms. Should I say that again? Regulation allows for a diversity of cell types in multicellular organisms. So cool. Now, chances are every cell is making some ATP synthase. So there are some genes that are probably being expressed in every single cell in your body. But not all of them. Oh, somebody had asked how many genes there were. And I looked up, the Human Genome Project estimates that there are between 20,000 and 25,000 genes in humans. Another cool thing, we have 23 pairs of chromosomes. So we have 46 chromosomes, and dogs have, I think it was 39 pairs. Wow, they have more than we do. Crazy, huh? Chimpanzees have 44 pairs. I know that because one time I put up a karyotype of a chimpanzee on one of my PowerPoint slides, and then I went, Hey, there aren't 23 pairs of chromosomes here. All right, cell cycle. So we've got this brand new cell. And it's going to have a lifetime. Okay? Now it's going to make another new one here. So look at the pictures, they're better. And there are 3 primary phases. There's G1, S, G2 of what is called, okay, I'm going to stop G2 there, interphase. Interphase is the time of the cell's life that it is not going through mitosis, or if it's a cell that makes gametes, meiosis, and it's not dividing. Now there are 3 parts to that. G1, the cell's just been made, it grows a little bit, and it does its cell things. It does its normal metabolic functions of that cell. In S,